Okay, today I'm going to give an abbreviated history of astrobotany, plant hormones, and radiation botany. And on each of these, I'll give milestones and key features. So to start out, I'm going to tell you a little bit about my project. So I'm researching how ionizing radiation affects terrestrial. And specifically, I want to look at morphological changes, stress responses, such as hormone levels or hormone changes, and genetic double-stranded breaks. So let me give you a little bit of background on that. So a lot of what we know in this field, or how this field started out, was actually in World War II in mid-1900s. And what gave us the ability to go on this kind of space and astrobotany type research started with the Soviet ballistic missiles. And what these were is they were intercontinental missiles that gave us kind of the firepower, so to speak, and the technology to have rockets to go to the moon, and that led to the space race. During this time, we also found out that we needed a way station between Earth and the moon and for longer expeditions. And that's how the ISS space station came about. And one of the things we wanted to look at when we go to these other planets, like Mars, was whether there was life or any ancient history or ancient proof of life. But these require long-term exploration. And while now we do have, say, the Curiosity rover in this top right picture, searching Mars for signs of ancient life, if we send humans to do this and to make habitation a possibility on Mars and even on the moon, one of the things we need to be able to do is produce our own food and to be able to go from seed to seed with our plants. And this bottom right picture is actually showing a fossilized bacterium from Mars that had impacted Earth and was found in Antarctica. So this was kind of proof of ancient life on Mars. And these are other clues that we're kind of looking at. So, an understanding of how to grow plants so that we can use them as a food source for these long-term explorations, we need to understand the abiotic factors that affect plant growth. So this gets us into our astrobotany. So here's just some of the abiotic factors. So atmosphere composition, and that's things like CO2 and O2 concentrations, radiation, dose and type, light, whether it's solar, otherwise composition, intensity, direction, water and nutrient availability, temperature, and gravity, or in the case of the space station, lack thereof. So let's look at some history and milestones. So we grew, well, not the Russians grew the first plants to be grown in space in 1971. They were also able to produce the first plant to flower in space. And in their early sets of experiments, they also found out that since there's no gravity, when you water plants in space, the water kind of sticks to the roots and it causes waterlogging. So what we found out from those sets of experiments is that you need very porous substrate so you still get enough air to the roots. And there were a series of additional experiments later that showed us how each abiotic factor contributes to how we can successfully grow plants in space. So astroculture in 1992 showed us that for the nutrients needed to grow a full cycle for a full growth cycle, you can actually just include that in your substrate, either liquid onto the substrate or as pellets. The Chromex, which we'll talk about that more or mention it again, showed that if you grow plants in a confined space, the air quality is very important. And they found out that with no ventilation, volatiles such as ethylene, which plants naturally produce, causes plants to abort seeds, and they don't produce any seeds, which becomes a problem when you're trying to go seed to seed, and you're going to need subsequent generations. Pesto was a series of experiments which tried to mitigate all these previous aspects of abiotic plant growth. And what they found is that if you can mitigate all those secondary effects of good ventilation, tested this with plant respiration and photosynthetic activity. 2014 was a big jump because it was the first hardware and set of experiments which had the explicit goal of food production. And so the idea was to essentially make a space salad. It wasn't until 2015 that Americans officially ate space-grown produce. We don't know if they nibbled here and there before, but they were very interested. Although the Russians apparently have been eating their own space-grown produce since 2000. In 2018, we were able to launch a new, it's called an Advanced Plant Habitat, APH, which now has the largest growth capacity and most automated controls of all the other options we have for plant growth now on the space station. And even though it's only about half of a cubic meter, it has 180 sensors, which include leaf and root temp, relative humidity, CO2 and O2 concentrations, far red light sensors, and root moisture. And here's kind of just an idea of what some of these looked like. So on the left is actually out of, it's the same one that was used, the Oasis. So the first plants grown in space, and this is now sitting in a Russian museum. The middle one is veggie, which was the first for growing food in space for the space salad. And then the right one is the advanced plant habitat. And this is super dwarf wheat. All right, so let's continue on to plant hormones. So there's 5 classical plant hormones, but really I'm only going to be talking about one, ethylene. Now, the typical effects of ethylene include fruit ripening, abscission, and stem elongation. But what I'm interested in is that ethylene is also produced as a stress and wound response. So this would include radiation wound responses or stress responses. All right, so this concept of chemical messengers and hormones, plant hormones, is not a new one. So when the Darwins, father and son, observed things such as phototropism or phototropism, plants responding to gravity and sunlight, they knew that there had to be some sensory mechanism within plants and also some messenger that was telling the plants how to respond to that. They didn't yet know that it was a plant hormone, but they knew that there was some communication, some messenger with it. But it wasn't until 1901 that we actually discovered ethylene itself. And that was Dmitry Neldrubov who discovered that. And what happened is he was growing these plants in the lab, and he found out that the gas lamps they were using as a light source to grow the plants caused plants to be very dwarfed and curled over. And so this ethylene gas was being actually emitted from the lamps. And so that's what was causing this. Although he didn't yet know at that time, the plants also naturally produce the same gas as it were. And then more than 30 years later, Dane identified ethylene as also a gas hormone, and the only gas hormone that we know. It was still 50 years later before we actually knew the biosynthesis and regulation pathways, which we now call the Yang cycle. And it's named after him not only because he discovered many of the components, but also the key regulation enzyme, ACC oxidase, which was previously unknown. And he also found that it's actually an amino acid methionine degraded that makes ethylene. So after this discovery of ethylene, remember we mentioned the Chromex experiments. And this is again where they found that you have to have ventilation and good air quality. Because this ethylene, these plants were stressed out because they're growing in space, and they were producing this ethylene, and the ethylene was causing, again, seeds to not be made or seeds to be aborted. So a plant could not make a full transition from seed to seed, or it would produce sterols. All right, so going on to radiation bodies. So radiation was discovered in the late 1800s. Curie, as a doctoral student, Becquerel and Rock were all three key players. And although most of the research done shortly after there was X-ray and UV vis light, once radiation kind of came into the public use, such as watchmaking, we began to see instances of radiation sickness, such as the radium. And these were women who worked in the factories painting clocks with radium because it glowed, and they were getting very sick. So the significance of this is that that is the first instance of a long-term radiation study, and it was the first time that biological organisms were studied with radiation. So in the middle of this, we also had, in World War II, the Manhattan Project and Cold War, which of course resulted in Hiroshima, Nagasaki, and shortly thereafter, we had our world's first nuclear power plant. And with these power plants came subsequent disasters. Two of the most notable ones were Chernobyl and Fukushima. Although just shortly after the first one was built, there was another one called SL1, which was part of the U.S. Army project that was only in 1960. But Chernobyl is still a very hot zone and has very high radiation. So what does this have to do with the rest of this research in space? Well, radiation is a very big player in the abiotic factors of being able to make plants that can grow and will grow, either in space or on space missions. So we have to understand how biological organisms respond to radiation. So let's look at some key features. So ionizing radiation, which is the most harmful high energy type, is generally associated with detrimental effects, such as chromosome breaks, mutations, even cell death. I want you to consider three concepts. The first one is hormesis, and that's illustrated in this top right corner. The idea of hormesis is that when an organism is given a stimuli at a low concentration, it is beneficial. So like in plants, the low dose radiation can cause the plant to actually grow better than a control. It can be more vegetative growth, it can produce more seeds, it can produce larger seeds. But once you increase this dose, it then becomes detrimental. So that's where you see your fluorosis, your loss of color, your necrosis, your cell death, and kind of smaller, shrunk over responses. So another concept to consider is adaptation. And a good example of this is in the pine trees in Chernobyl. So this bottom right corner is actually a picture of Chernobyl. You can see the plant in the distance in the upper left-hand picture, the left-hand corner of the picture. And in the foreground, you see this kind of yellowish, reddish section of trees. Now, when Chernobyl exploded, the radiation didn't spread out evenly. So this is what's called the red forest. And on the left-hand side of the image, you can actually see the line that separates where low dose and high dose radiation was received. So in this red forest of pine trees, they found that the DNA was highly methylated. And when they did studies, they did 10-year-long studies where they took control non-irradiated pines and put them in that zone, and vice versa, seeds from that zone they put in controls. But what they found is that trees only developed the highly methylated DNA when they were in that zone, when somatic tissue was exposed to a high dose of radiation. And what this means is that it was an epigenetic effect. It wasn't, these markers didn't stay on the trees between generations. So if you got a tree from this section or a seed from the red forest and you transplanted it to a low dose or a no radiation zone, it would not maintain those markers. And this was a form of adaptation, as these methylation marks allowed the plants to prevent recombination and make their DNA more stable and less prone to mutation. But again, it was an epigenetic effect that you only saw when somatic tissues were exposed. So, and then the third concept I want you to consider is phytoremediation. This is using plants to essentially detoxify an environment. So in one paper, they used sunflowers, and they found that sunflowers can actually take uranium out of water. In 24 hours, it can decrease uranium in water by 95%, which was a huge finding considering how harmful uranium can be. So what are the applications of this? So we found that marsh soil has all the basic needs a plant needs to grow. Of course, the environment's a little different. The radiation is different. There's a lot of other abiotic factors that play into it. But there is the possibility, and that once we can control those secondary factors, there's expectation that the plants can still grow. And so extended space travel is one of the applications of all this research. And then like on the space station on the right-hand side, that's not currently on the space station, that's potential plans for the space station. So that they can have a facility where they can supplement their diet increasingly with plants grown there on the space station. Phytoremediation, again, like the sunflowers, to remove uranium. They've also been shown to remove radiation near power plants. And plants can also be adaptive to climate change. So whether here on Earth, if we're having more radiation due to depleted ozone, or hotter climates, plants can be adaptive. And we can study those with various techniques. So I hope you enjoyed the presentation, and if you're interested in any of the particular images or studies, I included the references for you in the next 2 slides. And let me know if you need any more information. Thank you.